Previous films from the Division of Experimental Biology have illustrated various aspects of work on the biological effects of low temperatures and on the preservation of cells and tissues in the deep frozen state. One of the films dealt with the work of Dr. Audrey Smith on the resuscitation of hamsters cooled to internal body temperatures below 0 centigrade, at which temperature some 50% of the body water was converted into ice. Two other films dealt with the work of Dr. Smith and Dr. Christopher Polge on the use of glycerol to protect red blood cells and spermatozoa against the otherwise fatal effects of freezing and thawing, and with the consequent feasibility of storing these cells for many years at very low temperatures in blood and semen banks. Yet another film, made by Dr. Delphine Parrott, showed how the orthotopic grafting of ovarian tissue treated with glycerol and preserved in the frozen state could restore fertility to animals sterilized by X-irradiation. The film you are going to see now illustrates the work of Mr. Ashwood Smith on extending these experiments to the preservation of bone marrow and to the use of a new protective substance, dimethyl sulfoxide, which in some ways is superior to glycerol for the protection of living cells against the effects of low temperatures. Here is Mr. Ashwood Smith to tell you about his researches.
Bone marrow stored at low temperatures with the aid of glycerol has been used successfully to treat the severe marrow damage which follows the exposure of animals and men to X-rays. Recent studies with mouse marrow, in which dimethyl sulfoxide has been used as a low-temperature preservative agent, are described in this film. The bone marrow is obtained from the femora of mice by aspirating with tissue culture fluid. The marrow is then sucked up gently once or twice into a syringe in order to break up the cell clumps. Marrow can be examined by placing the cell suspensions onto microscope slides which have been coated with nutrient agar. Under these conditions, the cells spread rapidly and motility can be studied for periods of up to 5 hours. One of the methods of examination which has been used is phase contrast microscopy. Time-lapse procedures demonstrate motility of bone marrow cells. Motility is best demonstrated at 37 degrees centigrade, and the apparatus which you can see here has been designed at the National Institute for Medical Research for this purpose. The movement of these normal marrow cells has been speeded up about 60 times, and you can see the characteristic movement of the various cell types. Only mature myelocytes and lymphocytes move from point to point. The stem cells, which are important in combating radiation damage, show only movement of the cellular membrane. The myeloid cells in mouse bone marrow are characterized by hollow, ring-shaped nuclei. At the top of this picture, you can see a myeloid cell, a relatively mature myeloid cell, moving rapidly. Living bone marrow cells may also be studied by ultraviolet fluorescence microscopy. The cells are treated with a 1 in 20,000 concentration of the fluorochrome acridine orange, mounted on agar slides and viewed by blue-violet light. Acridine orange is taken into the cell in the living state, and the nucleus of the cell, which contains DNA, fluoresces green, and cytoplasmic granules fluoresce red. This is a typical example of normal mouse marrow examined using this particular technique. Here is a myeloid cell in mitosis. Marrow cells which are to be preserved are suspended in tissue culture fluid containing 15% dimethyl sulfoxide, sealed in ampules and frozen slowly to minus 79 degrees centigrade in the original inexpensive apparatus described by Polge and Lovelock. When they have reached this temperature, they are taken from the inner methanol chamber and placed in a mixture of CO2 and alcohol at minus 79 degrees centigrade. The canister is labelled and transferred to a storage chest containing solid carbon dioxide. Here you see a marrow sample being placed in the chest, where it will remain for 1 month at this low temperature. At the end of this period, the samples are removed and examined for viability. The ampules were rapidly thawed by immersion in a water bath at 37 degrees centigrade. Slow thawing of frozen cells is to be avoided. The bone marrow cells were then ready for biochemical and morphological examination. One method of examination which we have used in order to assess the viability of preserved marrow is to study the biochemical synthesis of lipids and proteins by these cells. Radioactive sodium acetate is added to cell suspensions. The cells are then incubated at 37 degrees centigrade. 1 hour's incubation was required in order to assess the uptake of radioactive sodium acetate into the protein fraction of the cells, and 5 hours incubation was required in order to measure the synthesis of lipids. The proteins and lipids from these cells were then isolated, and the radioactivity which they contained was measured in an automatic Geiger counter. The lipid and protein from these cells was plated out onto planchettes, and the trays containing the planchettes were then placed in the automatic Geiger counter. The synthesis of lipids and proteins by mouse bone marrow is dependent on the presence of intact living cells. Cells which have been treated with potassium cyanide or homogenized show no evidence whatsoever of protein or lipid synthesis. Radioactivity measurements indicated that cells frozen in the absence of dimethyl sulfoxide showed very little evidence of lipid synthesis. These results are shown in the diagram. In contrast, cells which had been frozen in the presence of 10% dimethyl sulfoxide showed synthetic rates approximately 50% of the control values obtained with fresh cells. Very similar results were obtained when the synthesis of protein was measured. 10% dimethyl sulfoxide again seemed to be the best concentration. Frozen cells were also examined by phase contrast microscopy. Cells which had been frozen to minus 79 degrees centigrade in the absence of dimethyl sulfoxide were completely destroyed. When, however, the cells were frozen to this temperature, in the presence of 10% dimethyl sulfoxide, many of the cells were intact morphologically and moved actively. This is a megakaryocyte you can see here. Here is a group of stem cells with a small metamyelocyte moving on the right of this group. Notice in particular the membrane movement of the stem cells. Here again is another sample of marrow which has been frozen to minus 79 degrees centigrade with sulfoxide and thawed, and many of the cells are intact and are moving actively. This is a small group of stem cells, and the myeloid cell is in mitosis. Frozen cells have also been examined by ultraviolet fluorescence microscopy. These are cells which have been frozen in the absence of dimethyl sulfoxide, and only nuclear debris can be seen. This is another photograph of cells frozen without dimethyl sulfoxide, and on the right can be seen the naked nucleus of a promyelocyte. The presence of dimethyl sulfoxide, however, protected many myeloid cells from destruction. The cells which you can now see were preserved at minus 79 degrees centigrade in the presence of dimethyl sulfoxide for 1 month. Many intact cells can be seen. We had demonstrated that dimethyl sulfoxide preserved the morphology and biochemical activity of marrow cells, which had been stored for periods up to 1 month at minus 79 degrees centigrade. It was now necessary, however, to test the ability of this marrow to prevent radiation death. Unless mice which have been subjected to a lethal dose of radiation are treated immediately after irradiation with viable marrow cells, they will die within 30 days. Mice which you can see here were rotated in the X-ray beam to ensure even dosimetry and were irradiated with 1,000 R of X-rays. Immediately after irradiation, fresh and preserved bone marrow was injected intravenously into these mice, and the number surviving after 30 days is shown diagrammatically. About 80% of the mice survived which had been injected with fresh bone marrow, and about 73% of the mice injected with marrow which had been preserved for 1 month with dimethyl sulfoxide survived. Leukocyte counts on the irradiated mice treated with different doses of fresh marrow and with a standard dose of preserved marrow cells indicated that between 20 and 50% of the myeloid stem cells were viable after storage for 1 month at minus 79 degrees centigrade. Since this work was completed, recent studies have indicated that dimethyl sulfoxide is superior to glycerol for the low temperature preservation of mouse bone marrow.